Welcome everyone to this ACCS and OHBM webinar and workshop series about the brain imaging data structure. And before we get started, I would like to acknowledge the country on which we're situated on and the traditional owners of the land. And I acknowledge the Turrbal and the Jagera people, which live in St. Lucia, where I'm situated on. And we acknowledge the traditional owners and the custodianship of the lands and pay our respects to the ancestors and their descendants. And what we have planned for this webinar today is we have an exciting program of multiple talks around the brain imaging data structure. And the format we're trying today is a little bit new to what you have seen in our previous OHBM webinars. We try a little experiment because we want everything quite interactive and really show how things are done. And so really, as I said, this is the first time we're trying this. Let us know what you think about this. So really, there will be a survey later where you can let us know, and I'll show that in a second. So we will start off this webinar with two famous international guests, Rémi Gao, and speaking about BIDS for fMRI, and Guillaume Niceau introducing BIDS for EEG-MEG. Then we have a short break, and then we switch gears to a more Australian perspective on BIDS, where we have Catherine Hall and Chao Su from the Australian Characterization Commons at Scale initiative, who show us how to access compute resources in Australia to work with BIDS. And after this webinar today, there will be small group hands-on sessions organized by ACCS, where you can actually do this and you actually get help in really applying this. So today is really the show and tell, and then there will be follow-up sessions. These follow-up sessions, I'll just explain that here. So they will be limited to around 20 people because this is about how many desktops and virtual machines we have to run this. Which one you sign up to depends on where you are. If you are on a Queensland university, you sign up on the one CVL at Wiener and BIDS at Wiener. If you are anywhere outside of Queensland, you sign up for CVL at M3. And the links just appeared in the chat magically, which is fabulous. So click on these links. And if you are interested to have a more detailed hands-on session where really you click through and you get help and you get stuck, then sign up for these. And they will be run by Catherine and by Chao, and I will be there as well in this. Then the next webinar is already planned as well, which is the 7th of December, a Tuesday, from 11 to 1 p.m. AEDT, so Sydney, Melbourne time. We also got the feedback, don't do it in the morning, don't do it in the afternoons. We're trying also to shuffle a little bit with the times. And in this webinar, the next webinar, we will show examples of research using collections, so large data collections like the Human Connectome project data, in how they do this in their research. And then there will be hands-on sessions for accessing and working with HCP data in the CVL, in the Characterization Virtual Laboratory. If you're interested in joining this, you can already sign up now. So if you click this UQ Zoom US link, this is a registration link for the next webinar in December. And then the next webinar topics are fully in your hand because we would like to know what you would like to see. So please fill out the survey now and shape the next webinar series. So we already have a couple of respondents there, but I would like to see basically everyone telling us what they would like to see, give us feedback on our current webinar series, what we did in the past, what we should do in the next weeks. So also this link should appear in a second in the chat. Good. So then I'll move on to introduce our speakers. So it's a big pleasure to introduce Rémi Gao, a research fellow at the University of Catholic, working on laminar fMRI and one of the BIDS maintainers. And we have Guillaume Niceau, a researcher at Indiana University and member of the BrainLife.io team. And Guillaume is the chair of the BIDS steering group and the lead of the MEG BIDS extension. Then, in the second half of the webinar today, we have Catherine Hall, Training and Community Engagement Manager at the National Imaging Facility and lead of training for the ACCS project. And finally, we will hear from Chao Su, Head of Technical Team at BrainPark, Lab Manager at the Neural Systems and Behavior Lab, and Data Scientist for the ACCS project. So I hope after this little starter, we are all hungry to hear about BIDS. So over to Remy, the kitchen is yours.
Okay, first unmute myself, otherwise you're not going to be able to hear me, and then share my screen. Otherwise, this is going to be not interesting for you. All right, let's do this. Cool. Can you all see my screen at the moment?
Yes.
Awesome.
Okay. Looking good, yeah.
Brilliant. Then I'm just going to post a link to, nope, to this, if I can find the chat. Where did it go? Chat, come back. Hello. All right, so that's the link to the cookbook for BIDS that I sort of put together for this session. And I've been spending way too much time on Twitch this last few weeks. So this is going to be sort of a live session of me trying to create a BIDS data set from scratch. And I want to be clear that I'm going to do this by hand, but in practice, you don't want to do this by hand. This is just to show you, sort of to give you some intuition of how BIDS is organized and what are sort of like the main concepts around BIDS. But yeah, in practice, you either want to script it or even better use one of the converters to convert your data from whatever comes out of the scanner, MEG, EG, into an actual BIDS dataset. Okay. And so in case you're not aware, let me just get to that reference. So BIDS is, the way I put it is, that's the Marie Kondo for fMRI, MRI, EEG, MEG, data curation in general, where you get from a whole bunch of data that's completely disorganized and you have no idea what is where and what it's named, what hides behind the name of the file names, into something that's like highly structured and that everyone sort of agrees on and you can sort of share easily with other people. The other thing that I need to mention about this is now it also supports PET imaging and also extended sort of anatomical data that were not supported until like a few months ago. And there are extensions in the process to sort of also support eye tracking data or microscopy data, or there's a whole bunch of what's called brain BIDS extension proposal that try to expand what type of data are supported by this. So if your favorite type of data is not in there, have a look at the extensions that exist. You'll actually, I'm just going to show you while I'm at it, where you can find that list of extensions. So you go on the brain imaging website and into the Get Involved section and scroll. And here you have the list of all the extensions, what their topic is, and you can just click on any of those links. And for example, you want to know what they are doing with eye tracking, right? And you can get it into a Google Doc where people are working on trying to expand the BIDS proposal and try to sort of see how that type of data is supported. And there you can just sort of even participate and extend and give your opinion on what that type of data should look like when you wanted to have it into BIDS. Okay. Okay, so let's just jump right in. So here, this is going to be just a practice. So we're going to be using some source data that actually come from one of the SPM dataset that has a whole bunch of like EEG, MEG, and fMRI data. I'm only going to show you how you convert the MRI data. What you would technically need to do this would be just a file explorer on whatever computer you're using and a text editor. In this case, I'll be using Visual Studio Code. I tend to recommend one of those three. In general, you want to use a proper file editor because it can help you just deal with some file format more easily, right? And sometimes you might need to convert file formats. So in this case, I'll be just using MATLAB and SPM to show you how to do it. Once again, if you're dealing with real like fMRI or MEG or whatever type of data you're using, use a proper converting tools to do this. That will definitely make your life easier. All right, so I'm going to be doing this pretty much all in Visual Studio Code. And I'm literally going to go through this. Okay, first we want to create the folders where we're going to put our data, right? So let me show you how I would do it. So first, that's how I tend to do it. You create a folder for the raw data, which will be where your BIDS dataset is going to live. And then in there, you can put a folder called source data, which is where you can put all the data that you are going to try to convert, right? So this is, and BIDS doesn't tell you how that source data should be organized. And I'm just going to put my source data in there, right? Okay, cool. What is the next step? Yeah, okay. You can any code you used for the conversion, you can also put in there. So we don't have actual code. What you can do though is put the content of that cookbook into there so that people know what kind of recipe you follow to do this. I'm gonna get the raw content of this to do it. Instead, let's do this. Let's add a folder for code, and in there, one for conversion. And in there, we just want to create, I don't know, a README file. Oopsie. Oops, obviously, this is too early in the morning for me to be able to type. So there we go. Good, that's better, right? And I'm going to put that there. So now we have the recipe as well as here in case you want to sort of know what you're supposed to do. Good. Okay, now we got this. Another thing you could do, for example, is that you can also here store the code that you use to run the experiment. That if you have functional data, like you can put the code for like, you know, whatever it's like SciToolbox, PsychoPy, whatever software you use to run your experiment. I do recommend you also put your code in there. And another thing you can do is also have a folder with your stimuli. That's also completely allowed by BIDS, and you can put your stimuli that you use for this experiment in there. Cool. Okay, what is next? So we have our folders. Okay, so we have the data for one participant. So we're going to need to create the folder structure for that to like host the data for that participant. Okay, let's do this. So the way you do this in BIDS is anything that's a subject folder as the sort of prefix, and then you have a label for that participant. Labels can be anything with like letters and numbers, it's fine, but no special character in them. Then in there, you can have what's called a session folder. This is optional, but in this case, it can be useful because we have MRI, EEG, and MEG data, and we suspect that those have been acquired on a different day, but how you decide to organize that session folder is pretty much up to you as a experimentalist or data curator. It's up to you decide how to do it. Let's create a session dash MRI folder. So we use the prefix ses for session. Okay, and then in there, that's where you put the different folders that are going to host the data coming from different modalities, right? And so we have anatomical data in this case. So we're just going to call it anat because that's the word we use in BIDS for anything that's like structural data. And then we have another one for bold imaging, and that is going to go into a func or functional data. You could then have the one for MEG, and like they're pretty self-explanatory, but each of those type of data is going to go into one of those folders. Okay, so we have our folders. Okay, so now we need to convert the structural data that we have. And it's in our source here. Obviously, I messed up the imports, so you should be going back in there. Good. All right. This is the anatomical data that we have. So very often when you have neuroimaging data, it comes out of the scanner, and very often you're going to be doing what's called DICOM data. Here, this is a bit different. It's an old data set from SPM, so this is a file format you might encounter, but more if you going to dig into old data set. I'm also doing it here with this type of data so that you can see that BIDS can sort of like adapt around whatever input file format you have, and you can still sort of do it. But there is something a bit specific about BIDS that when it comes to neuroimaging data, you want to use a nifty file with an extension that goes .nii. And so this type of format is not supported by BIDS, right? Where you have the image and the header about the image in different files. That is not supported. So we need to convert that thing. So let me start MATLAB. And then we're going to do it with SPM. But this is also to show you what you sort of would have to do if you had DICOM data. You would have to convert your DICOM data into this nifty format. So this is a bit similar to what you would have to do anyway. SPM also has functionalities to sort of do that conversion from DICOM to nifty. Here, I'm just going to be doing something a bit different. Okie doke. I should have prepared some jokes for the waiting times. Well, better luck next time. There we go. Okie doke. All right. So when you're in SPM, the way you would sort of do that conversion, if you have to do it through a GUI, would be like you go into SPM, where is it? Util, and then you go with 3D to 4D file conversion. And then you just go grab the file that you want to convert. So it's in source data, structural MRI, and that's the file you're interested in, right? And you want to make sure that you've got the same data type. Yep, okay. And then you just run this. And another thing you want to do, I suggest, is everything you do in general, just keep track of it. So, so save that batch that you just created into the conversion, so that in case you go back to it later, you can always sort of know what you did. Okay, so now we should have our little conversion scripts are there, so we know what we did. That's good. And now we have a 4D file.nii. This is what the thing we're looking for. Okay, so now let's move this into the ANAT folder. And now we need to name it. So we need to give it a proper BIDS valid name. So that's where I'm going to tell you a little bit about how BIDS files are named. So there are a few things you need to do about a BIDS file. So first of all, it has an extension. So the thing that comes after the dot, so .nii in this case. So, you know, like all files have extensions pretty much. Then what comes right before the extension sort of tells you information about what type of data you're dealing with. And we call it, in the BIDS world, we call it a suffix, right? And in this case, it's a T1 weighted image. So we have T1 weighted, right? So it's going to be the suffix, and right before the suffix, you have an underscore. And what comes before that is a series of what we call entity dash label pairs. Oops, label, right? And you can sort of chain those one after the other as long as you separate them by underscores, and then it's fine. Then it's always entity dash label something, right? And then you can have as many as you want. So this is how you would sort of construct a BIDS file name, right? Obviously, there are some constraints on what the entity names have to be for a given sort of data type, for like a T1 weighted image. And if you want to know what entities you can use, all right. So this is what I was supposed to give you. Okay, so if you're interested in sort of figuring out what entities you can use for a given data type, so this is the BIDS specification. So I'm going to show you how you would get there. So this is when you get into this BIDS specification, this is where you're going to land, right? This is just, I wouldn't call it the Bible of BIDS, but this is like the reference we go back to if we want to sort of figure how things are supposed to be named or organized. And anything that has to do with a specific modality, imaging modality, are going to be into the modality specific sort of chapters. And then here we're going for magnetic for the MRI page. And all of those pages, if you look at a given section, will give you like templates of how things are supposed to be named. And it will tell you which entities have to be there. Those that are optional are between like square brackets, and in which order they're supposed to be like chained one after the other. And as you can see, when it comes to anatomical data, there's a lot of things you can do. But in this case here, we are going for T1 weighted images. So that's like the general rule that's going to apply is like this one. So, and you don't need to use all of them. As I said, anything that's between square brackets is optional, so you don't need to use it. Let's keep it simple in our case. And the thing we need to have is the subject entity and the label that we can find in the folder name. So it's going to be just sub 01. Then you have the session. We're using a session folder, so we need to have this in there. So it's going to be session-MRI. And then we can leave it at that, right? It's just all the other entities are optional, so we don't need to have them. So that's all we're gonna have. So now we can just rename our file here. Okie doke. So nope, there's an extra extension that just came in here that I don't need. There we go. Goodie. All right. So entities required. Yeah, they must come in a specific order. Okay. I think, yeah, so far we're doing good. The next thing you're going to want to do, and you're going to want to do that very often when you're dealing with BIDS dataset and you're creating them, is use the BIDS validator, right? So this is just, it can run in a browser. It can also just run in on your like machine locally if you don't want to use the browser, but it's fine. And the thing that it does is that it's just going to look through your files and in the BIDS folder where you're working and just check that the file names are okay, the folder structure is okay, whether there's any file missing. So it's giving you a feedback on how you're doing, if you need more stuff, or if you can maybe add a little bit more of this, a little bit more of that. Cool. Okay. So where did I put this data again? Right there. So oh yeah, and one thing just to know is that your data is never uploaded, right? So don't worry about like privacy issues. That's it. It only checks the file names and the folder structure. So it says upload on the button, but it doesn't actually upload anything, right? But it gives you a report on what it did find into your data set and whether there's something that makes the data set not valid, or whether there's some things that could be added, right? So in this case, it's telling me you're missing a dataset underscore description JSON file, and you could add a README. All right, so what is this dataset description.json file? Okay, so that is, let's go back to the BIDS specification. Nope, wrong tab. All right, in case you're lost in the BIDS specification, just Google it in the search bar, and then you should be pointed to where you should go. So every BIDS dataset must have one of those dataset description.json file that contains just metadata or like information about your dataset. If you're not familiar with JSON files, they're pretty much just a style of text files with a very specific format. But if you can just create them in your like file editor, not an issue for this. And let's just create it here. And the only things that are a bit, they have their format then with like a lot of curly braces because it comes from JavaScript. And what information you're supposed to put in there is explained in this table, saying like, okay, these information are required, and all the others you can put, but it's just it's recommended. All the others are like optional. One way you can create them is either by sort of grabbing the content of one of those examples from the specification here. For example, you can just copy this and then add it in there. So, and then you can just change the content of those fields to just whatever you need for your use case. Another possibility that you can do is use the templates from the BIDS starter kit. So there is a BIDS starter kit that tries to sort of help people figure out how to just create their own data set and deal with BIDS in general. And there is a folder called templates where you can just have sort of empty files like this that you can use to sort of create your own like data set JSON or whatever. And so it would be the same here, and then you can just fill in whatever you need. And the two only required one were name and BIDS version. And I think now the latest version of BIDS is 1.60. And name, we're just gonna go BIDS cookbook, just because I have no idea how to name it otherwise at the moment. Cool. Okay, now we've added the dataset description that the validator was asking about. Let's just check whether this is making the validator happy. Okay, upload again. Oops. Okay, now we've got more errors. Oh, that I love when that happens. So now we have some invalid JSON file. Okay, so apparently the template. Okay, so ethics approval should be an array and dataset type should. Oh, okay. So there's some information that actually should be filled in, right? So dataset type cannot be empty. So it has to be raw, for example. It can be raw, or the other possibility is derivatives if you're dealing with data that has been already processed. So that's another part of BIDS that I could tell you about, but that would take much longer. But here we're dealing with a raw data set that has not been pre-processed. And what was the other one? Ethics approval, supposed to be an array? I don't remember. Memory span. Oh yeah. Well, one thing I'm gonna do right away as well, just so that you know how to do it as well, is that I've just used a template that came from the BIDS starter kit, and obviously that template is giving me errors. So let me just open quickly an issue to tell me and all the other maintainers that dataset description template is invalid. Didn't work. Did not work in the BID validator. So that is the problem I was gating. And if you want to be a bit thorough, you can even just do a quick copy paste of this and give us more information so we can help you in the future. That is technically, if you open issues, supposed to be doing that a bit in a cleaner way, but I don't have the whole time to just go through the whole thing, but at least that will sort of give you give us some information on how to help you later. Okay, so are things better now? Are we doing better? Do we have fewer errors? I want fewer errors in my life. Yay, okay, so now we still have a warning. That says you could add a README in there. That's a good point. Let's add a README. Okay, so templates. We also have a template README that I do recommend to use because it's pretty much has a huge checklist of all the things you should put in a README. So let me just grab this and put it in the dataset. Right, README. Cool. Okay. So, and then obviously you would have to fill in that README with the actual information that concerns your case, but at least now you have like, some sort of template to sort of get started from there. And hopefully now we should get no errors. Yay. Okay, cool. Brilliant. What I like to do as well is just grab the content of that little table and put that at the top of my README so that people, when they open their data set, they know what's in there. Cool. All right. How are we doing? Okay. We adding the missing files. Oh, yes. So we can add some metadata to that file. So the idea is that every file in BIDS dataset can have a JSON file that goes with it, where you can put information about that file, right? So, and it's all in a JSON format. So we're going to create a new file, and it's going to be a JSON file. Okay, so there is actually extra information about the dataset I didn't mention or forgot to mention because I'm not quite awake yet. And it should be in this README. Yes, exactly. So let me grab the content of that README and put that into our source dataset as well. Okay. New file, let's create a README in there. Oops, there we go. Cool. And Guillaume and I sort of extracted some of the data from the SPM manual and other places where we could find information about this. Yeah, and this is the information about the anatomical data that we can add into the JSON file. Okay, let's see how we're gonna do this. Let me show you how you would do it with the templates. No, actually, I'll do it like by hand fully so that you see how you would do it if you have no idea about the templates first. Cool. So you would have to sort of go into the section about your that concerns anatomical data. So MRI's page and then anatomical data. And then, oops, nope, sorry. We're dealing with metadata that's actually common to a whole bunch of MRI stuff. So it's going to be in the common metadata field. So that's why we use templates, because even I get lost in that specification, and then it's easier to have a file where you know what you're supposed to put in. So these are the name of the metadata fields you can use for MRI data. And as you can see, they have like very specific names. And so that's how you would do it. So in general, we use, oopsie, hello. Okay, then we're gonna do it the hard way. In case you're never sure how they're supposed, which case they're supposed to use, it is, it comes from JavaScript. So you're gonna be using what's called camel case, right? Where the first letter of each word is like, has to be uppercase. So you get used to it, but at the beginning you're like, why is it like this? Well, there is a reason behind it. Then we had the manufacturer model name, which is the type of MRI scanner that they used. Okay, yep. And then there, oops. Okay, and then field strength. Where are you? So yeah, it's magnetic field strength. That's how you would do it. Cool. All right. Yeah, you can imagine that if you have many files, you don't want to do this by hand, clearly. But at least you sort of see this way where things are supposed to go and how you name things. Cool. So now we have a JSON file for our anatomical MRI, and things should still be valid. If they're not, oh, okay, we're good. Brilliant. And next thing I wanted to add was where was I? Participant.tsv. Yep. So this is where you put information about the participants of your data set, right? And TSV stands for tabulation separated value. And you could write it in like directly in the editor here. I want to show you can also just create them with Excel or whatever sort of software you're using. Oopsie. To deal with tabular data. And how are they formatted in case you're confused, once again, you can just go participants, files. Okay, so it's participant.tsv. And there's an example at the bottom. So the required column is participant ID, and then you should have the name of the participant, sort of the entity and the label of the of each participant under that column. And then you can add like extra columns to this. So let's grab that first one and take it from there. Yep. Of course, that's not working. All right, let's do it by hand. Yep. Participant underscore ID. And you can have an age, for example. And we have one participant at the moment, and which is just sub-1 and age. We don't know. So when you don't know, just put NA. And we can save this as a TSV file. I don't know exactly how you would do it in Excel, but I think it would be fairly similar. Cookbook, and we want to put that in there, and it's going to be participants.tsv. TSV. Yep. Nope. So yeah, that's the tricky thing. To actually save it as a TSV, you need to save it as a CSV, but then you tell it that the field delimiter in between columns are going to be tabbed for tabulation, right? And there we should be, and then you need to rename it because it's taking some liberties on how it names things. So yeah, okay. So now we have a participant.tsv file, and you can sort of see it in the editor because there's an arrow between like the different columns that tells you that you're dealing with tabulations here. And another thing that's annoying is that I'm pretty sure, not sure whether the validator is going to catch this one. Yep, it is catching this one. Participant ID must consist of sub-yep. So the problem of using Excel is that it did put an uppercase on that first letter, and now this doesn't match the name of the participant. So you need to sort of change this, otherwise it's not going to be happy. So one of the problems of using Excel is trying to be too smart. Okay, now we don't have any error, but now it's telling me that it's actually good to have a JSON file to go with that TSV file to say what's in it. Okay, for the moment, that would take a bit too long, so we're going to continue with this. One thing you can do, and I like to do, is also use DataLad. I'm not going to explain too much about DataLad because that would be an entire workshop just by itself. But what I like about DataLad is that it's a bit like you can create checkpoints on your data so that if you just mess up something and you want to go back to a state of data that you had before, you can always just sort of travel back in time, provide a sort of like safety net where you can just like be crazy, go wild, but always come back to something where you had a clean data set. And you're gonna use tell DataLad to create a DataLad data set. Use the force because there's already data in there, so DataLad would otherwise not create data set. And you need just to give it some configuration information. Okay, DataLad just creates some extra files in there that you don't need to worry about at the moment. And then you just need to make a first save with a little message so that you remember what that was about. Okay, and I kind of like doing that at irregular intervals during when I'm creating data, and every time I have a BIDS valid data set, so that I can always go back to a state where the data was the data set was valid. Okay, then the next step is actually just converting the functional MRI data. So it's gonna be very much the same. So I'm just gonna do it a bit more like rapidly here. But the idea is that we are gonna want to sort of go and grab, we're gonna want to go and grab our functional data and convert it to a 4D format. Nope, now was the right folder, but it's in here, and we go for the functional data for, there were 2 sessions actually. That's a terminology issue. So you select all the files because this is what's called a 3D format, where each image from your time series is a different image, is a different file. And the interscan interval, I remember from memory, which is like your repetition time, is 2, but I've written that somewhere. And then you just convert the file. And hopefully it shouldn't take too long. There, done. The issue then is that you don't need to go and grab that file. There it is. And then you put it into your func folder. And then you need to give it a name. So, all right. So it's going to be sub 01. We are dealing, it's still the session, oops, sorry, MRI. You need to use a task entity to say what the people did, and they were seeing faces in this data set. Nope, it's a dash, I mean, not an underscore. And then this was the first run of a 2-run session. And then you use the suffix bold because you're dealing with bold data, and then you have the nifty extension. So this is how you would construct it. Oops. Wow. Okay, so this is your first run. The second run would be sort of the same. Actually, I forgot to save this, so let me just save that quickly. Oops, code conversion. No, but here, this is the func. Okay, good. Where was I? There. So now we need to get the second session and do the same. Let's save that as well, just so that we don't mess things up and we can always know what we've done. Good. Oop, hello. Where did you go? All right. That's the second session. Doop. There, done. So session 2, that's there. Yep. Let's just put it with its friend here, there. And this is run 2, so we can just pretty much use the same name, but change just the label of the run entity, and we have our 2 runs. Okay, how are we doing in terms of BIDS validity? Are we still on track? Are we missing something? Okay, okay, so what are we missing? We've got some errors in here. So, okay, we need to have information about the repetition time and the task name. That is because you need to have data set, you need to have JSON files to go with those files, right? Where you put .json, and in here, whoopsie. So you need to have the task name. Oops. I remember those because I've had to type them many times, but you would have to sort of look up what the actual metadata fields are for these things. And I'm pretty sure that we also have repetition time, just like this. And it was a 2 seconds repetition time. Cool. Let's create a copy of this because we need one for the run number 2. Up. Okay. And okay, now we're good. So we just have a few warnings. Slice timing information. We tried to look it up, could not find this one. The one that's actually important is event.tsv. To analyze your functional data, you're going to know what happened when. So what stimuli was presented at what time. And to do this, I could do it by hand, but that's going to take way too long, and it's not going to be fun. So I have a little script in there to do it, and I'm just going to sort of use this, sort of show you how you will try to make your life easier if that becomes necessary. Right, so it's just this function. Let's just go grab the file. It's in source and it's in functional MRI, and it was the trial session. Oops, and obviously that doesn't work. Yep, it doesn't work because I'm missing some things in here. I just need to add this to the path, and let's do that again. Yep. Okay, source, functional MRI, fMRI, and yep. So it should have converted it. Where do you put it? Doop, doop, doop. Oh. Now I cannot find the file anymore. I love when that happens. Oh. Yeah, there it is. Sorry, my bad. And then let's put it into our folder with there. And you would need to give it an actual proper BIDS name. And it would have to match the session it corresponds to, right? So I accept that we're dealing with an events type of data type. So you would just call it events.tsv. And this is just a table file that just tells you when something happened, how long it lasted, so the duration, and what that event was. And this has how you would organize your data set. I mean, sorry, your sort of onset files.
Remy, we're slowly running out of time.
Yeah, I know.
You think you can wrap it up there?
That's it.
So that's already
it. Yeah, yeah, that's pretty much it. Because the rest is just a little extra that you could do, but I don't want to get into this, so I'm going to stop it there. Because then the next one is like you do the same for the next run. Let's just make sure that we end on a good note and we don't have any errors, and then we're good. Yep, no errors. That's what I like. Okay, sorry, it took way longer than I had planned, but I hope that was somewhat informative.
Yeah, thank you very much. So yeah, it was really cool to see. Really, so this was basically BIDS by hand, right? So no one would actually do this for a big data set, but I think we put it in the session because we thought it would be really cool to see what's actually under the hood, and that it's nothing scary, right? It's just text files and naming files. That's what BIDS is. And later in the second part of the webinar, we will hear also how to do this automatically, right? There are tools to do this. So don't worry, you don't have to do this, but I think it's really, really helpful to understand what's happening under the hood. Cool. So with this, we're moving on to BIDS from a different angle. So now we looked at functional magnetic resonance imaging, and now we're looking at MEG. So Guillaume, the stage is yours.
Hello. Can you hear me well?
Yes, we can hear you perfectly.
Okay, so thank you, Remy, because the good thing that Remy has gone through all of this is that now I can go much quicker. And even though that we don't need to do everything by hand, it's super useful to know how to fix the errors, to know where to look when something is wrong, or how to add some details that are missing. So now for MEG, I'm going to, let me share my screen. I will also, I will also share my notebook here. I will at some point put it on GitHub, but I couldn't do it for now, so you can look it there. And I'm going to share screen, the whole desktop, just in case something Yeah, okay. Can you see it?
Yes.
Yes. Okay. So I'm going to use the same dataset that Remy was using, but the MEG side, because this is a multimodal dataset. And to look at the MEG specification, you can look at the webpage that Remy was pointing at before, which is this one, where you have all the template or how to organize your files, how to name them, and all the details that should be included in the make JSON sidecar JSON file. And it's very useful, as Remy was saying, which fields are recommended and which ones are required, so you don't forget any of those. However, in this case, in MEG, we have to be careful because some of the MEG files cannot be renamed by hand. Which is the case for example, CTF files, which are DS folders. So there are some tools that can help you rename the files automatically for you, because they contain some files inside the DS folders that cannot be renamed. Yeah, that you have to pay some attention. So the example, there are the different toolboxes, MEG and EEG toolboxes have some functions to deal with this. So MNE Python has the MNE BIDS tool, which is the one that I'm going to use first. And there's some documentation there. Then in Brainstorm, you can use through the user interface. So it's really easy as a first approach. And you don't need any coding. And it's also for MEG, EEG, intracranial. And this would be the second example I will use. And there are other tools like Filtric Function Data to BIDS and EEG Lab for EEG data BIDS MATLAB tools that you can also use in the similar way. So let's start with the MNE BIDS. So first, I have synchronized my Google Drive here. So it's already mounted, so no worries. Then you have to import some of the libraries and install MNE and MNE BIDS if it's not already there, and import them. Well, I must acknowledge the work of the MNE team, because they are doing an amazing team, and the MNE BIDS is also a really, really nice tool. It's super well documented. So in case you really have some questions or want to do something additional to what I showed today, you can just go there and see the tutorials. They have many tutorials that you can download and just run by yourselves. So here, the first thing we need to do, I have organized my data here. This is what you can download from the SPM webpage. So it's in the folder MEG. This is how it came. And this is what's inside. So you have some DS folders from CTF machines and a bunch of other files there. So the first thing you have to point is to a DS folder, to your MEG files, and then you can just read them with the MNE tool, MNE function to read files, CTF files. So it's stored now in the row variable. So there's some information that is not already gathered because it's not contained in the DS folder, like the power line frequency, which is 50, it was recorded in UK, and some others that you can look there that are maybe not included, and you can add them like this. And once you have your data, your raw data complete, then you may need to establish your BIDS path. For that, you need an output path, which is the BIDS dataset, or you can put the name you want the folder to be named. Then MEG, because the data type is MEG, and your subject would be subject 1, for example, the task is phase symmetry, or Remy was saying phases, so let's keep the same naming. And as you may see, for this run in SPM, they specify that they have 2 sessions. So these 2 folders are 2 different sessions, not 2 different runs. So in this case, we are going to just put run none, because it's just 1 run, and session, this would be session 1 that we have just loaded, is phases 1. So here then you will create your BIDS path, and I'm going to remove all folders that were there, because before I had a folder here that was called BIDS dataset that now will be removed. It takes a while because it contains a lot of files, but probably if I refresh, it may have disappeared now. What? Not now, not yet. I can do it also manually. Okay. And these lines, I've just only put this here to show you that sometimes MEG files can have calibration files and crosstalk files. This is not the case in CTF, but often is the case in Electa files. Sorry. So it's good that you know how to load them and how to add them also to the folder. So finally, you will have this many bits, write row bits, where you just specify your raw files and your bits path, and then it will create the folder for you. Here it gives you some summary of what the files contain. For example, the make site JSON file, make sidecar JSON file contains tax name that we have specified, phases, manufacturer, CTF. The power line frequency that was missing before is 50 Hertz, and sampling frequency. All this is contained already in the DS file. So it just reads and put all this without you need to write it manually. And then it also creates a channels TSV that is summarized here, and a few other like files like the scans TSV and all this. So if we go there, we see that a new folder was created with the dataset description files that Remy was explaining before. With this, the participants, because this information is not contained. This is the description, participant ID or age. This is just the description. This is the real data, and because subject 1 is not anything is specified, it just created with NA. So if you have the information, then you can populate these files. And the README, and as Remy was saying, it's important to know what's contained in the dataset. So then we will have the first session, make file, the scan session that is extracted from your file recording. Then is there the acquisition time that has to be input in this specific format, and inside the MEG folder you will have the session, the task for MEG, and the coordinate system JSON file, channels TSV, events TSV, and the sidecar make JSON file. So so you have a look of what's inside. You will have your MEG coordinate system, the units, city and centimeters, head coil coordinates, nation, LPA, RPI. All this is containing the data, and many bits just extracted from it and organize it according to bits. Also, the channels TSV will be automatically created with all the information about the channels and all this. So this is super useful to do things manually. So you can print the bits tree, the directory tree, and see how everything is organized inside. And also you can see your events here. Because we didn't touch anything, we just have the events that were contained in the data. But you can also add, as Remy was showing, a TSV files with the events, and also pass it as an input to MNBIDS, and it will mark everything and synchronize everything accordingly. Let me show you. Here you just to say events data and events ID, and this is events ID is just a dictionary with the events. For example, trigger 1 is faces, trigger 2 is scrambled faces, and all this. So it organizes it there. And finally, well, not finally, but maybe you want to mark some channels as bad. If we read our BIDS dataset from our new BIDS path, that the one that we have just created, and plot and print which are the bad channels, there are not any bad channels. There are no channels marked as bad currently. So we can say, okay, we know that these 2 channels were bad in this dataset, in this recording or in this session. So we can run this, specify there the channels that we want to mark as bad, and then just run MNBIDS dot mark bad channels, and then you put in the channel names the ones that you have indicated, and override true, then run, and you will see that this is, if you run this again, then you have to read again the data file because it's saved, and then you run, and you have 2 channels marked as bad. If you made a mistake and you want to reset all the channels and say, okay, no, all my channels are good, then you can run this, and all the status of your channels are marked as good now. And again, if we upload the data and we just print the bad channels, we'll see that now there's any bad channels anymore. So sometimes you miss some fields that you want to edit some fields or add some additional metadata. So this is also possible here. You just have to get the BIDS path. Now it's important to add that you want to, for example, if we want to the MEC JSON file, it's important that we specify the suffix, which is MEG, and then we'll get a BIDS path that we can make a copy and say, okay, and specify just that we want just the extension JSON. So our new sidecar path will be this one to be edited. And then you can add as many entries as you want. For example, if we want to add the power line frequency because before we have forgotten or because we have entered it wrong, and it's 60 hertz instead of 50, then we can add it here. Or the manufacturer, instead of CTF, it's Megin, then we can add it here, or we can just go there and add as many files as we want, as many fields as we want. So we just update the sidecar JSON file and pass the sidecar path that we just created with the entries, with the new entries. So this will create a new JSON file, as you can see here, with the data that has changed. You can also do it like this if we want to return again manufacturer CTF or power line 50. So you can add more or modify whatever you think. You have to be very strict in the naming because this is what is going to be written in the JSON file. So if you have a spelling mistake here, it won't pass the validator later. You will have an error. So as Remy was showing you, you can just pass the validator. I have downloaded the dataset before, but you can, well, Remy has just shown. Yeah, this is the MNE dataset that we just created, and we just say upload, and well, we have a warning, which is like we have very few authors in the dataset description. So we'll need to go there, and as Remy has showed you, add a few more authors, or just programmatically write the authors in a complete way here on your code. So that's the example for using many BIDS. And now I would like to show you also how you could do it using Brainstorm. So maybe some of you have already used this toolbox. And if that's the case, I'm going to delete this project, sorry.
Guillaume, just quick, there's a question in the chat from Simon Vogrin. Can BIDS be used to override any parameters within MEG files? So it is data, sampling rates, etc., or is it exclusively to document what is reported by the original data format?
The default outputs, it reads what's in the dataset. So if it's a DS folder and has a specified sampling rate and all this, it will read it and it will be the default. But if you want to change that, you can do it manually on your code. So let me see the chat. Can it be used to override any parameters within the make files? Well, BIDS is a standard to organize and rename your files. So I don't really get the question of to override any parameters within the make files. You can do it, and you can report it on BIDS. Because BIDS is just starting to organize your files, how you name it, how you document. So yeah, BIDS is not a tool to modify files inside. Yeah, you can use tools like MNE BIDS or MNE, and you can modify your files or resample, downsample, and all of this with your usual toolbox that you use to analyze your data, to treat your data, but then you have to report it in BIDS.
Cool,
so,
thank you.
Yeah. I don't. Yeah, I don't know if it was clear, but otherwise just ask me again. I don't usually look at the chat, so let me find, because I have all the controls down here and I cannot see. Okay, here it's MATLAB. Okay, so we can create a new protocol, which is, let's call it 2021 workshop. Brainstorm. So default anatomy, no, using the, because here I don't have the MRIs, but you can just in the default protocol anatomy. And in case you want to also upload the MRI, you could do that. But for now, let's go like this. This is the new protocol. So as BIDS call it, and then you would like to add a new subject. We can leave the name here because this is a non-BIDS dataset. Any non-BIDS dataset that is imported in Brainstorm can be exported into BIDS. So here we can leave a random subject name. And then we'll need to upload the files. So if we say review raw file, in the same way as you may do before, this is the MEG folder that we had, and we just import the two CTF DS folders. So this will be as you usually process your data or load your data. And once you have them inside, we have one now. Sorry, my computer is a bit slow. Okay, because this is not the MRI, maybe it doesn't fit very well. The real MRI, maybe it's inside doesn't. And also the alignment, you can check. This is very good. Brainstorm allows you to align very well and see how the cap is aligned with your data. So that's what you have there. So then you will have here your files. This could be any protocol you have already created before. So if you just drag and drop your files here and click run, you will see how you can, in the file screen, tab, say export dataset. It's experimental for now, but I think it does a good job for a first approach. So you have to specify your, I have the old one, so I'm going to delete it because I was testing it before. Okay, so let's go into Brainstorm and let's create a new folder. I'm going to say BIDS Brainstorm. So here, this will be the folder I want to use to store my data. So Brainstorm, BIDS. Oh, sorry. Not double clicking. Select. So you can select subject names because you can batch this through like many subjects together. You can just get numbering this or custom names. Here I will leave the numbers. The same for the sessions. Let's keep a number. And if we had empty room or like this, it will also go to an additional folder that will be called empty room as BIDS specifies. Define MRI before export because this is a default. We don't need that. Overwrite the system files. Well, we could have used this one instead of deleting the folder, but power line frequency 50 Hertz, make position DWAR during the scanner upright. And if you want to add a few more things like project name, you can have this OHBM dataset or project ID, description. You can fill all this as you like. And then you will click run. And you'll see that it exports here. Well, it takes a while because it's long. It's heavy. We're writing MEG files. You see how it's starting to create the things. You have your dataset description that you for sure will need to maybe tweak a bit or edit a bit. License name, BIDS version, authors. Here you may need to enter a few authors, otherwise you will get an error in the validator. And you will have your subject. Inside your subject, you will have session 1 and session 2. And in session 1, you will have your MEG. In the same way as MNBIDS created the scan session, here it's also there. File name, and this is the file name and the acquisition date. So you will have it here on the scans TSV, and then you will go to the MEG, and you will have this run first session, run 1, and the make subcar with some of the task name. Well, because we didn't specify any task name or task description, it's generic, but then you have your manufacturer, CTF, sampling frequency, power line frequency, DWAR position, digitized landmarks, false. Well, I think they were there, so maybe this you can populate them, or this could be updated so it will also copy here the digitized landmarks and head points because I think they were there. I'm not pretty sure. And the spatial grading compensation. So this is work in progress. People at Brainstorm are trying to get updated with the new specifications and every update of BIDS. So probably if a field is missing in the same way as Brainstorm and as MNBIDS, if something is missing you can just open a pull an issue on GitHub and say, okay, my data contains these files or this data or this information, and it's not extracted on the sidecar JSON file. And we can work on it and see how can we fix it. But for now, all the required files are there. And if we go to the BIDS validator and browse the one that we have created now.
We're running again short on time a bit. Are you almost at the?
Yeah, I just wanted to pass the validator.
Oh, fantastic, cool.
Yeah, so you will see that we don't have any warnings and that we have all the no author. So you just go to the README and add the author. So as you see, all these tools that are automatically run can give you a very good approach of all the required files in a very fast way and smart way. So with this, I finished.
That's awesome. Thank you very much. That's really cool. We will do a 3-minute pit stop break just so that everyone can get quickly to the toilet and then come back. And then in the second half, we'll look at compute resources, how to run some of these tools automatically on infrastructure in Australia from Catherine and Chao. So I'll see you back at 5:20 in 3 minutes, and then we'll start there. So yeah, thank you so much, Guillaume and Remy, for your insights and for showing us really hand by hand. That was really, really cool.
Okay, one question. The questions on the chat, because now I see the chat and I see that there are...
break, exactly. So yeah, whoever wants to run off quickly, then do this. And otherwise, yeah, let's answer the questions on the chat.
Or maybe we can do it after all the workshop is finished and we can just...
If you can answer it now, that would be great. Yes, no worries.
I didn't follow, so I don't know if is there anything that is very...
Okay, so I think one question that came up a little bit is EEG. So MEG and EEG, is it quite related in the BIDS standard? Is it fairly similar to go from what you just showed to an EEG data set?
Yeah, exactly. Yeah, I didn't go to the EEG, but it should be the same. Brainstorm, in the same way as you import your EEG data, then you can export into BIDS in the same way, just clicking the same buttons. The content may be different but some of the parameters are shared, like sampling frequency is the same for MEG and EEG, and some specific fields, like for example, manufacturer cap or cap manufacturer or EEG reference, this would be specific to EEG, but it will be done in the same way. BIDS is also able to handle not only MEG, but EEG and also intracranial EEG, the same as Brainstorm. So all these three modalities will go very, very similar. And yeah, someone was asking to share the code, and yes, I have shared it before, but on my Google Drive, but I will post it on GitHub so you can just run it.
Yeah, and can you post the link again? That would be great. And then there was one more specific question about events files not found. Maybe you can just answer that in the chat while we go on with the next thing. That would be really cool. Fantastic. That's cool. Good. Yeah, so one more minute and then we'll continue with the next set. Catherine, would you like to start sharing your slides? And then I hope that was enough time for everyone to get a quick break, something to drink, quick toilet break. And then, so now we hear about how you can actually use resources to process data with BIDS. And this is presented from Catherine. So I think looks like everyone is back. So yeah, the stage is yours. And looking forward to hear what ACCS is.
One moment. I'm in immaculate timing. Just one moment. Hello, everyone. Yes, and as would happen, that was just my son. I'm sure you can all relate. I'm sure you're all similar to us, just doing lots of things at the moment, but never mind. Anyway, look, thanks everyone for sticking around. In this session now afterwards, after Remy and Guillaume have given us such a fantastic and comprehensive introduction there, we're just going to really quickly run you through what project the ACCS is, so why you're hearing from us today, and what the CVL is and what that means. And then also I'm going to show you very briefly how you can log in to a CVL session. And after that, Chao's going to run us through how once you were within a session, you would actually start to be interacting and using the BIDS modules within there. So again, I will put it into the chat later on, but the workshop registrations are open, and I would encourage you, please come along tomorrow if you're in Queensland and you're able in Brisbane to come to the session. It will be on in the Sir James Foots building, and I can send you the exact information, but it's room 341. And I'll also be hosting that over Zoom, and at that session I will run you through the steps that you need to go through in order to set up your access. And on Friday, we'll have a similar session for users in other locations around Australia, so not Queensland, to access another cluster. And I'll explain the difference very shortly. And then that will set you up ready for next week, when Chao is going to go through in great detail how you would work with each of those modules within the CVL system itself. So if you come tomorrow or on Friday, then you will be all set up and ready to go next week for the intensive workshop with Chao. So just make sure I'm on the right screen. Here we go. So yes, so today we're going to talk a bit about what is the ACCS and what is CVL and what is the difference and should you know, and what does all of that mean. As Stefan said, my name is Catherine and I am the Training and Community Engagement Manager with the ACCS project. I work for the National Imaging Facility and I'm based in Brisbane. If you need to ask any questions at any time, please always email me. My email, I answer it pretty quickly, I hope, but it's catherine.hall@uq.edu.au, and I'll put a link up for that later on as well. And I can help set you up with CVL and get you in touch with people who might be able to assist with training you on any topic that you'd like to know about. So I'm just going to basically just quickly give you an overview of what the landscape that we're working with in the ACCS project is actually like. And I'm sure most of you are probably familiar with this sort of situation, which is just that there is just masses and masses and masses of data being generated, and it's only increasing every year. And the number of instruments are increasing, massive instruments. And now not all of these instruments have got the capacity to have persistent identifiers, but that is increasing as well. So we're able to track metadata with the data coming off machines, as well as these machines are producing data in all sorts of different formats, and getting them to be interoperable is quite a challenge. So these are the sorts of challenges that everyone in the imaging community is facing at the moment. It's not just brain imaging, but I'm sure that's what's of most interest to you guys today. And essentially we can see here how the challenges are quite universal for all imaging scientists, but that they interact in different ways. But there are some definite common and shared challenges there. And some of those are basically that, as I said, that the rate and the quality of image acquisition is increasing exponentially. And unfortunately, we're not necessarily always able with our own laptops to handle all of the pre and process and post-processing at a large scale that would be required. There are also challenges with capturing metadata and making sure that our provenance is good so that data become trusted. And ultimately, as with all scientific domains, we're trying to increase our fairness and trustedness with everything that we do, and do this along repeatable and reproducible pipelines. An additional complication for you guys, particularly in working with the sorts of data that you're working with, is that a lot of this data can be sensitive and needs to be de-identified. And so these are challenges that the ACCS in particular is trying to help to work with you to find solutions for, and pathways where you can overcome some of these issues. So ACCS itself stands for the Australian Characterisation Commons at Scale. And the aim of this project is basically to, as I said, address a lot of those previous issues. And the way that we do that is by bringing together some IT specialists and engineers to create a particular environment, so a work environment where you are able to go from acquisition through to final deposition of your data through the analysis steps in between, and do that in a sort of contained and repeatable as well as accessible space, which is free. And helps you overcome some of those problems, particularly with handling sensitive data. Now, within the project, we are working with a number of communities, the large and correlative microscopy community, and a special interest to you would be that the work we do with human bioimaging. And as well, the project also works with the scattering technologies communities. Each of these domains is a specific work package within our project, and Work Package 5 is the project that is dealing with human bioimaging and hosting of collections and reference data for you to work with for your experiments. And ultimately, we're offering a federated solution that you can access with your AAF logins, so institutional logins if you are an Australian researcher or affiliated with an Australian Research Institute, and run this under a common banner. So how it differs to the CVL, oftentimes CVL, ACCS is used interchangeably, but CVL is just one specific tool or platform that we work with, which helps to offer some of these solutions. And CVL itself stands for Characterization Virtual Laboratory, and it's our biggest product and our main focus for a lot of our work at the moment is the CVL. And like any virtual laboratory, there are other ones you may have heard of, genetics ones, but this one is specifically for imaging sciences, and it brings together workflows and tools within the one package and location that you can access, and it's free and will help you to complete your analysis. It's accessed over the web, and essentially it's a graphical interface for you if you want to use Linux tools, and the aim of it is to reduce the burden on you to know any coding and also help with bringing your data into one centralized space and analysis environment. The other thing that's really nice about CVL is that it is built in Australia specifically for Australian researchers. So the point of that is that if there are any engineering issues, some IT advice that you would like, people are actively working on this, and they're very accessible and very quick to engage with you to come up with new solutions or add features that you may want. So it's highly responsive and adaptable to your needs. So the major benefits of interacting with CVL and why I would encourage you to sign up and get an account is that it will make software easier for you to access. It's free. It's all been pre-installed for you. You won't have to do any compilations of your own. It's a low-code environment, not no code, but very low code. It allows you to reproduce your pipeline. So if you've used a particular version of a piece of software, they will remain in CVL in perpetuity and be continually supported, so that you may have done half of your analysis in one version, a new version's come out, but you don't want to repeat the whole analysis, so you can always go back and get that original version that you've been working with. It also, the feature of it is that it will allow you access to GPU processing. So it's situated on HPCs so that you can go in and have access to much more GPU than you would be able to support on your own laptop. It's also integrated with other repositories and collections like CloudStore or institutional collections and other data collection and data management tools. It's integrating XNAT for human bioimaging, and so it will make collections available to you within that space, so you won't have to download them again and re-access them. The other thing about it is that data files can be transferred quickly, and that using tools like Globus, perhaps for very large data sets, which would be checksummed at the end so that you can be assured of their integrity and also of their security. And you would have to move your data a lot fewer times. So making here. So the use cases, and there's quite a number of different communities that engage with CVL, but in your case, the main thing that would be here would be this for processing large collections, and it would be the graphical visualization that it offers using large amounts of GPU, which may otherwise be inaccessible to you. In the case of Weena in Queensland, the CVL node here, you can access a huge range of tools, particularly, and we have a very well-developed neuroimaging suite there. Thanks, Stefan. Stefan's responsible for a lot of that, and we thank him very much for that. So I'm very quickly just going to show you what it looks like in practice, because that's just who knows what any of that really means. So if I get out of there and stop sharing that one, and I'm going to share this one here.
Just wondering, will Shao that show as well?
I think he's going to work within it. So I'm just going to quickly show you how to log in and what that there are a number of options. Chow will work off M3, but I'm going to work just show you how to log in. So you would go to desktop here, and then in Queensland you would use Weena, and in other locations you might use M3. And it's quite easy. You just select the node you want there. Can you still see this login at CVL?
We can see the browser and we can see logging it in, yes.
Okay. So I'm going to go Queensland. No, not QUT. No. Okay. It's 2 ahead of myself, the University of Queensland. Continue. So it uses the AAF logins. So anyone who's got an institutional account anywhere can just log in using the same access there. And again, you go through your MFA.
Dr. Just I can quickly show that part.
Okay. I have to.
Yeah.
That's how you would set up a desktop. So it's really quite easy and very accessible through a web browser. So I will go stop showing that. It's just going to do it. So I'll go back here, share very quickly. So all I really wanted to say was that just as a little graphical summary of how you might work within the system would be that you would take your data from your instrument into a repository, which would then be accessed by CVL itself, and then push it back out into other stores. And in the workshops, we'll show you a little bit more of this workflow. But the whole purpose of this is to increase your fair approach, a more fair way and trusted way of handling data. So just very briefly, as I've said, the main thing that we're interested in the ACCS project in doing is creating a secure and pre-configured environment that you can work with, which will allow you to have reproducible pathways and allow your research to be accelerated. We also offer training, which we hope that you will come and join in, and also suggest anything that you might like further knowledge of. And there are the workshop things. I'll put them in the chat, and I'm not going to waste too much time on it, and I'm just going to let Chow, then he'll log into a session and show you how you would then engage with the BIDS modules from within the environment itself.
Fantastic. Thank you very much, Catherine. So yeah, that was an overview of what ACCS and CVL is. And now we move to the last part of this workshop. So we have about 25 minutes left, which is a little bit less than we had planned. So Chow will show a BIDS conversion from, again, another level, right? So in the beginning, we saw very manual, how to understand everything, and we saw the MEG BIDS. And now we see a quite automatic way using a tool called BIDScoin on the CVL. And everything that Chow chose today will be gone through into this hands-on session. So the second session of these, Chow will actually go through this. We have 2 hours time. You can play with all of this yourself, make mistakes, and we can help you to get this done. So we will probably start with showing how to get access, so you see a little bit what's involved, and again, just skip over things so you see a little bit what's coming in the hands-on session. Okay, Chow, with this, the stage is yours.
Yeah, thanks for the introduction, Stefan. Just on top of that, you can even bring your own DICOM data and you want to play with it. So that will be really cool for the hands-on session. Thanks for the great presentations by Remy, Juma, and Catherine. Thank you very much. So I'm just going to start these interactive sessions. I know we're running out of time, so but still I want to use the first 1 or 2 minutes. I want everyone to be a quick survey. So type in these 3 numbers. If you are not not know about a lot about neuroimaging, and particularly in the MRI, just press 1. And if you have neuroimaging, especially MRI, but you process things on your local server or on local computer, press 2. And if you are a user on CVL, no matter is it CVL Massive, Monash, or CVL on Winners, press 3. So I will just take a 1 minute. I know it's out of time. And another thing is, Stefan, please, because I can't see my clock very well, 2 minutes behind, just stop me. That's okay. I'll stop. As Stefan said, everything we're going to resume and it will be revisited later. Just a little bit while I'm doing that, it popped up. So myself, my name is Chow, as introduced by Stefan, so working in Monash University. Basically, I'll just say, my experience of neuroimaging is from number 1 to number 2 to number 3. So now I'm using CVL for like 8 years now. I'm just very happy with it. And then just quite happy with all of things. And today is going to show you one of the tricks that just easily convert DICOM to bits using these coins. I can see lots of people put in 2 and 3, and then it's really good. And probably someone's in the 1. Ashamed to what's a bit shy. But that's okay. I will try to stay in the middle ground just to cover everything. So if there's anything that's missed again, so everything will be revisited. And I will just start it now. So firstly, I want to touch is why we want the bits. So from an imaging processors, researchers' point of view, so I was asking why this is important. So as we know, the bits is from a DICOM, from the scanners, you get the DICOMs, and then you convert into bits. I always make this comparison, the bits like a key. So they can actually open another doors. And then the doors means all of these powerful pipelines, and then also the big community behind it. And then once we have the data in the proper format, and then we can do a lot of things. At the end of the day, you're probably saying, we can generate some beautiful pictures, and that's good for your publication, etc. So that's just a simple why we straight away on here. So today we're going to base on the CVL. As you say, people have their own way to do it. I see lots of people typing 2, so which means that you probably have their own way to convert it. And as Remy, it's really hardcore. They can even type in everything by itself. That's really cool. But today I got to do a lot of automatic. And then again, so this one, I was thinking it's good for the people in 1 and 2 who is just starting the imaging journeys. And then I want to really know a little bit how this data is organizing and what you're going to do with it. But if you already like a hardcore is working on imaging for a long time, this is just another way of converting bits DICOM to the bits format. You probably find it's interesting. So I'll break down mainly into 3 sessions. The first one is the examples of we got one set of DICOMs and how we convert it to bits on CVL. Simple as that. And secondly, we will extend it into multiple participants and multiple sets, because it's very unlikely you're going to have one person in the project, right? So how are we going to apply this one to process a batch of participants DICOM to bits? And third one, I'll just touch a little bit of the benefits of bits and how it is connecting with what I said. It's function MRI, fMRI prep, MRQC. You will see, as long as you get everything sorted, it's just like one comment away, one line away from your bits format to those beautiful pictures. Okay. Amount of time, let's just quickly go through the first one. So firstly, I prepared a lot of the data. So all of the data I prepared as a practice data and we copy into the folder. We're going to go through that slowly later in the hands-on session, which we got 2 hours to go through this. So we'll be fine. So, but once it unzipped it, so here's the file structures. I want to just talk a little bit before the interact session. So this is the way that I like to organizing my file structures, but everyone have their own way to do it. So simply that the first level is projects. And in here with the first example I'm going to do is, and then in the second level, there's a lot of folders in here. The DICOM DR is usually, I call it DICOM DR. That's what the DICOM file is. And then here, each participant will have another subfolders in here. And the parallel to DICOM there is the BIDS. I call it BIDS. Someone called it raw date, and someone called whatever, nifty, whatever you like. And then that is the BIDS structure it is. And then there's the .ini, the JSON file, etc. And then let's just focus in these two folders for now, for example one. Okay. So once you unzip the data on in the hands-on sessions, you will see the structures of this. And then, so I just take a screenshot of what is the DICOM DR looks like. This is probably remind some of the unpleasant experience when you get some data from the scanner or someone give it to you from, no matter if it's a hard drive, it's a CD-ROMs, you find all of the DICOM is stuck in one folder. It's like you go home with all of the pages of several books, but you don't know which page is going to which. So that's what we're going to start. The first part of the BIDS is called the DICOM sorting, which is going to do that. That's going to the interaction mode now. So, which is I'm going to quit, and I still open my slides like that. Just for that, I will quickly popped up my slides in. Yep, I've just put my slides in the chat, so everyone can have a look. If you miss anything, so you can always check my the slides shared online. So here is the folders. If you open it, so the example one, and you get a DICOM, and then in here, that's all of the DICOMs in there. So the first thing we want to do is want to load the software called the BIDS. There's a few ways to doing it, but I'm going to use the hardcore way, which is using the terminal. So to do that, I go find out one of the in the project level folder, and I go to File, and then just open the terminal. So I got my terminal out here. You don't really need to be, if you are new to terminal, just don't be really scared of it. So you just type in something, and then the computer will react. So I'm in the So I said I'm in the project directory, and then here's the command I need to type in. So I put in here. The first step is DICOM sorted. So module load BIDS. This is the first line, which just means I'm installed this software to my background. You can imagine all of the software is preloaded. It's like a bookshelf, and then terminal is your desktop. And I always say, when you module load, you grab a book from the bookshelf on your desktop. Now you're working on it, and you can module unload, put it back. So it's quite simple. So there's a peace of mind of you reinstall a lot of software. It's going to be headache. Okay, so the next one is just BIDS, just called the DICOM sort. So what I'm going to do, DICOM sort, and then I'll point it to exactly that folder it is. It's a subject one, which is all of the flyers of the books are all mixed together. So here's the directory. I pointed to that folder, and there are some options. Which just means like rename it. You can just remember what it is, and when you type in that, it will sort it. And you will see there are some subfolders created, and now 496 files is reducing the number, and then they will chuck it into the books, and then they will line up properly, and then staple, and everything's ready for you to do the next step. So while it's loading, the amount of time, I just quickly go through. So after that, yes, you will see the end product will be a few of subfolders in here, which is the DICOM goes to where it should be. And those subfolders is actually the modality, right? So in here, we're just doing the typical structure and the function MRI. This is a task-based function map. And then there is a field as a field map, or this is something that we will need to leave it out as an example. So the next one is called, the step 2 is simply called the BIDS mapper. I like it pretty much because they will bring a GUI rather than we're just working on the terminal or code. So this will give you a little bit relax. So just type in this command, BIDS mapper, as this is the name, okay? So it's just doing mapping all of the DICOM to the BIDS format. And then just tell it exactly the actions and the settings we need to do. Okay, so I press that, and it will load a little GUI here. So here, so you got everything, all of the information you need in the GUI. So firstly, you will need to, so this is a special problem for all the CVLs, you need to change the path of DICOM to NNX. As you can see, the DICOM NNX is the backbone of BIDS. I know quite a lot of people probably just use DICOM to NNX itself. Totally fine. So this is just another way to do it, as I said. And you can click test it, everything works. Now go to BIDS format. As I said, this is just dealing with the 3 subfolders within DICOM DIR. And then the first one is you can tell MP2 range, that's a T1. Now you can edit the action. So click edit. On this side, you can see all of this information that's directly extracted from the header of the DICOM. So thank God, those things will be extracted automatically. Don't need to type in. But I like is this part. So you can tell this what modality it is. So I know this one is structure, so it's anatomic. And then you got a choice to put a suffix. You got a T1, T2, etc. So this is really cool. And then you can just say, if you know what it is, you just put it on there, and output name will be generated automatically. Of course, you can always changing this, but that's related with the flags of the DCM to NNX. If you want to play with it, you can do it later in another tab in the hands-on session. Click OK. And then this one is the reference image. So I pretty like this function because sometimes I have some of the DICOM folder that I don't want to convert. So I can use this function called the leave out. So that means when I convert to BIDS, it will bypass these folders and you don't need to worry about it. And the last one is the finger tapping. You can see from the name, the run one, it's definitely a bold, which is the function MRI you put on here, and the suffix is bold. You got other options in there. As long as you know what to do and then happy with it, and you do is save it. So this is our draw attention of this. You save into a file called the BIDSmap.yaml. It's in the folder called the code and the BIDS folder. So you will see this is actually quite powerful. So you can reuse this information so when you batch a series of participants and you want to convert it. Okay, so that's it. That's done. Second step, exit. All right, so let's go into the third step. So to make it a little bit interactive, I go to the BIDS. So we see this folder, you can see that folder, the code folder is there, which is the YAML file, and the my Nifty files and my BIDS, all of the should be generated here. Okay, so I'll skip all of this. You miss anything, don't worry. So all of this will be in my slide. You can just follow it. I'm sure that's awesome. Everything is fine. So in the step 3, another thing is I organize my slide in the way I have the $, which is after that is also the command that you can just directly copy and then paste. Edit paste. Hope it's working. Oh, it is working. So it's a simple just BIDS coiner, which is the command, and the input DICOM DR, and the output is the BIDS. Okay, you can see it start to generate something. It is working. I got my README, got my data set, description.json, yeah, all of the other files that we stacked during the BIDS validating of the warning should pop up. Yeah. So while we're waiting, I just quickly showing visualizing what it looks like in here. In these levels, in the BIDS levels, you will see the subject. Oh, it's done. You see subject 1, and also in this level, you will have all of these files that you need, like the participants' TSV file, and you can see the subject IDs, and then there's the genders, and then age, weight. All of this is extracted from DICOM, and also for some reason, when I validate against the BIDS validator, it's stuck in the participant JSON file. But if you remove it, that's totally fine. But it's not stop you from other process within the MRQC, etc. In here, in subject 1, you will have anatomic folders and functional folders, and that will have the Nifty files and then JSON files in there. As you can see, JSON files are all of the header information, metadata, so saved in the way that human can read, and plain text file. Okay, so I will, because of the amount of the time, I will skip all of the visualization things. So just basically calling FSIs to check it. And then we'll show that in hands-on sessions. And also you can refer to my slides to see what it is. But I do want to draw your attentions over here. So it's this one. So in the codes, in the BIDS, the YAML file, yeah, that's what we talk about. Here is all of the information that you remember you changed in the GUI. So everything is here, and you can even change in here just instead of plus the GUI. And also draw attention, the last bit of things, they do accept the power file, which is from Philip, and then P7, which is from GE Scanner. So if you want to bring your own DICOM, you want to try it, just do it on the hands-on session. Okay, so I think I'm pretty much done for this. Yeah, now I'm going to the second example. The second example is I got 2 sets of the data. So once you use my the prepared data and unzip the example 2, when you open it, you will see this. So you go to DICOM folder, you got 2 folders, right? So you open it, it's still the it's gonna come 496 data. 496 DICOMs in there. Okay, so let's forget about this one. Let's start a new one. So again, I'm in here in the example 2. Open a new terminal. Okay, I'm going to do exactly the 3 steps that we need to do before. So firstly, module load. Yep. And then, so here is, instead of I'm going to, this is slightly different. I'm going to type in this. So when do the DICOM sorted? It's still DICOM sorted, DICOM sort. Instead of I type in to specify which participant it is. So I'm just saying, go searching all of the DICOM DL folders, right? And the -R, -E is the exact same flag as we saw before. And then here is the difference. So it's a subject prefix sub. Yeah, it's pretty straightforward. Just say all of the folders with the prefix of the sub, they'll be considered as the folder they need to process. And they should recognize as there's 2 folders and convert it for you. And later on in the BIDS, right. So let's have a look at it. So it will go through that pretty quick. And I'll just prepare for my next slide. And it's going to come very soon. Yeah, so it's working on subject 2, which is very soon. It's going to be coming along. Right. Yeah, all done. It's pretty fast. And then the next one is here. We're going to take advantage, that I said, we take advantage of the YAML files here, which is the one we saved before. That's a configuration of all of the settings. So I always do this practice. I create a new folder. Remember that I have a folder called the scripts. Yeah, I like to put some of the things I was referring into that folder, make a copy, but you can also point it to this folder directly. So this is the old YAML file I have, and a copy. And I quickly switch to the scripts, and I just dump it here. Okay, now I'm back to my BIDS, and then let's get ready. So what we're going to do is we just type in this line in here. It's actually very similar as the one with before, only except they have a flag called -B, and then to point it where is the YAML file it is. Now press enter. You will know what's the difference. This is the same. See. Remember, this is all the red font now become green font. So they just copy exactly what is your procedures before that you set up. So everything is here, and you check this is all happy, and click save, and you confirm and consent. Just use this YAML file. I will update it, and now file exit, and you can just run the last bit, which is this pointer, which is convert. Let's say DICOM dir to BIDS. Done. It would take a couple of seconds.
While this is computing, we have a quick question in chat. What modalities can BIDSCoin convert?
That's a good question. So I tried function MRI, structures. Are you here probably referring to EEG, MEG, those kind of things? Haven't tested yet. I'm sorry, I'm doing a lot of neuroimaging with this.
Yeah, so as far as I know, it can't do it yet, but this is something that people are working on and making it more general so that you can also do this for EEG, MEG. And yeah, I hope this is coming soon. But currently, as far as I know, it's not possible.
Yeah, again, the backbone is the DCM to an NX. So it's that one that's been changed. Then whatever that was covered probably isn't covered. Okay. All right, I think that's probably, while it's waiting, I'm just quickly go to the next sessions. So I've got a question before asking how this is actually working. All right, so you will see from here to next, before the one line, to kind of solve it, is we need to create a folder called a derivative, which is the, simply you can think it's the output, all of the things is in there. So, okay, perfect, just finished. Okay, so what we can do is let's just keep going from here, create a folder called, oops, sorry, it's not here, I'm in the wrong folder. Yep. Great folder derivative. Okay. Okay. And then I copy and paste. I have prepared some script for everyone. In the script, there's one for FMI prep and one for MRQC. And let's just say copy. Come back to here. Again, I put everything in the script. If there are script, paste in here. Okay, so that's it. What we're going to do is let's have a look at MRQC. Double click on here. I want to use my favorite. Okay, so you'll probably find that this is a little bit busy if you're the first time doing the coding. But actually, firstly, the blue font is the blue lines. They are just the there's a comment. You don't really need to the computer is not going to read it. So the first part you always will see is module load. It's the installed software that you want to do. So it's MRQC 0.0.7. It is one of the old versions. I just showing as example. And here is the variable design. It's like if you use the MATLAB, etc., they just assign some information to a variable. So that's subject one, and that's a variable. And then here you just need to have a working directory. This can be pointed to anywhere. If you forget about it, it just pointed to a work directories in here. It's called a work. It's random. And then here's the most important thing: is the input of MRQC. It's actually exactly the output of your BIDS folder you created. So you're done. And output, you can accept. That's just the folder you just created. And then the last thing is you need a license. So for now, you don't need to worry about it. You can just borrow the license we have for the hands-on sessions, but eventually you'll get your own. And then there's just one line code here. If you can read it clearly. So this is the MRQC -v is for verbal. Just print everything onto the screen and then tell exactly what is the participants you're going to run. So in this case, the subject 01. And then you have working directories, which is that input, output, and tell them participant level. And you just as simply as run it. If to run a script, just bash the script, the MRQC, and press enter. It will run. The amount of time,
Yeah, we're almost out of time. So I guess it's great you got to this point. So if you just want to wrap up, and then we'll ask one more burning questions, and then I think we'll call it the day.
Yes, exactly. Thanks. So because of the time, I'll just quickly show you what is the output this looks like for the MRQC. So derivatives are run before in the QC group, and here in the script T1. So you have this beautiful, because I only n equal to 2, and then you will see all of these parameters on here. So, and you hover your mouse on it. This is picture subject 2, subject 1. You'll find it identical. Yes, I just duplicate the first one as the second one. You can tell. Yes, so that's everything. You click on it, it will navigate you to the individual reports, and those are the beautiful pictures that you're talking about. You want to get. So probably it's just exactly same thing for function. So FMI prep. I'm sorry, there's a question about FMI prep in there, but it's clearly in there. It's exactly the same. You just bash the script I gave to you, or you can just run the script. Just the last line of the script. If you really want it, just join the hands-on sessions. I'll put it in here. And we got time. I'm gonna help you hands hold everyone step by step to go through here.
Okay, fantastic. Thank you so much. And sorry for cutting you a bit short, but with this, we're perfect in time now. And the good thing is everyone can really go through this in the hands-on session. So make sure to sign up for these sessions if you want to play with this interactively, and then you have the full attention of Chao and Catherine and really 2 hours of time to go through it and really learn how to do it. Are there any burning questions before we wrap up for today? Yeah, otherwise I think the chat was quite busy, so that's really good. Looks like lots of people got great answers. I will put this recording on YouTube, so you can look at that afterwards. Again, there was a lot of information in this. I hope it was useful. I thank all the speakers for all the work they put in preparing this and presenting it. And I hope it was really good to see BIDS from these different angles, and also good to see it all interactive and not just as a slideshow, but actually seeing how people actually do it with everything involved, even a live issue that was filed on Git. So that's really cool. So yeah, thank you very much. And then we are looking forward to see you at the next OHBM and ACCS webinar in December. And then hopefully, fill out the survey so we know what topics you want to see next year. So thank you very much. See you soon.
Ciao, everyone.